So we can get started. So what we're going to do today is we're going to start talking about the vasculature, and most of the time in here has been spent discussing the pump, the heart, which moves the blood through the vasculature. But there's some ideas I want to get across about the vasculature before we leave the cardiovascular system and go on to blood and immune. And one of the ideas I'm going to try to drive home here today is that this vasculature is a closed system. I'll show a couple of slides to that effect. That is, the blood never really leaves that closed compartments of a series of tubes once it comes out into the aorta on the left side or out into the pulmonary artery on the right side. But we will see that there's a tremendous amount of branching, that the single tube, whether on the right side or left side of the heart, is carrying blood away from the heart. But that single tube then branches and branches and branches, and particularly on the systemic side, that aorta is going to branch into a number of major arteries that then are going to branch into more arteries. So you have this tremendous branching system in such a way that we're delivering that blood out into the tissues. And finally in the tissues, this branching is going to end up in the capillaries. And the capillaries are going to be the exchange vessels. These are the sites where oxygen is going to leave the blood vascular compartment and go into the tissues. It's the sites where the carbon dioxide is going to leave the tissues and go into the blood. It's the site where nutrients, such as glucose, are going to be leaving the blood and going into the tissues. All of that occurs across these specialized vessels called the capillaries. The capillaries are said to be the exchange vessels because they are so thin, they have a single layer of endothelial cells, which will allow movement across from the lumen of the tube across into the tissues. So we'll talk a little bit about that system. And then we're going to talk a little bit about the autonomic control of the system. And the autonomic control of the system we're going to emphasize is going to be the arteries and the arterioles. So we'll see that the single aorta branches into arteries, further branches into arterioles, and these further branch into capillaries. But along this pathway, all along this outward pathway of arteries and arterioles, we're going to see that those tubes are surrounded by a layer of smooth muscle. And that smooth muscle layer is under the control of the autonomic nervous system. The autonomic nervous system in this particular case is going to be the sympathetic branch of the autonomic nervous system. And the sympathetic branch of the autonomic nervous system, as we've seen before, is going to release norepinephrine. The norepinephrine is going to bind to a particular kind of norepinephrine receptor called an alpha-1 adrenergic receptor. Now, if you get down into the weeds of all of these adrenergic receptors, there are 3 betas and another series of alphas. There's an alpha-1, alpha-2, there may be an alpha-3. And each of these do slightly different things. But the one we're talking about today is alpha-1. Alpha-1 is an adrenergic receptor. It's going to be on the smooth muscle of arteries and arterioles. The activation of these alpha-1 adrenergic receptors leads to constriction of the smooth muscle. Now, if you imagine the smooth muscle winding around the blood vessel, if you contract that smooth muscle, that's going to reduce the diameter of that tube. Basically, if you imagine having a hose and you had some constrictor around that hose, you'd reduce the diameter of the hose. Well, that's what we're doing here. And why that's important is back to Poiseuille's law. And we described Poiseuille's law when we talked about the coronary vasculature. But the long and the short of Poiseuille's law is that it says that the flow is dependent upon the pressure differential times pi times the radius of the tube raised to the 4th power, divided by 8 eta L. Well, again, we'll simplify this way down. 8 is an integer, we don't have to worry about it. Eta is a Greek letter standing for the viscosity. The viscosity of the blood is going to be the same wherever we look, so we're not going to mess with that. L is the length of the tube we're thinking about. Well, we're not concerned about the length of the tube so much as we're concerned about its diameter. The pressure differential is just the pressure differential along the length of the tube. Pi is a constant. R is the radius. The long and the short of this, what this is going to boil down to, as we said before, is that the flow is going to be proportional to the radius of the tube raised to the 4th power. That means small changes in the diameter or radius of the tube is going to have a big change. These small changes in the radius of the tube will have a big change on the flow. And we'll talk a little bit about that. And one of the ideas that will come out of this is that by changing the diameter or radius of tubes in this highly branched system, we can now redirect blood to different places. So by constricting a portion of the tubular system, we can redirect blood other places because the resistance to flow is going to go up so fast with the radius. Doesn't take much in the change of the radius. We have a big change in flow. I'll try to walk you through this sort of intuitively as well. And then we'll talk about blood. We'll start down the road here of what's in blood, start down the road of the blood and immune system. And when we break down blood, and we do this all the time, if you go to the doctor's office, the phlebotomist comes out and takes an aliquot of blood, they'll take that blood to a centrifuge and spin it. And when they spin it, they separate the blood into three major components. One component is going to be the plasma. The plasma is 55% of the blood volume. The plasma is basically just a protein solution. Basically, we're going to have 90% water, 8% protein, 2% other stuff. That's the plasma. It's the least dense. That's why it floats to the top of the tube. The most dense material will be the hematocrit. These will be the erythrocytes. And the reason the erythrocytes are going to go to the bottom of the tube when we spin it is because the erythrocytes are much denser than either the living cells in the buffy coat or the plasma. And the reason the erythrocytes are so dense is that they contain lots and lots of iron. We're going to see each erythrocyte contains about 250 million copies of hemoglobin. And each of those hemoglobins contains 4 atoms of iron. So each erythrocyte contains 1 billion atoms of iron. Well, that makes those erythrocytes more dense than most other tissues. So when we apply the force to the tube, the heaviest stuff, the densest stuff goes to the bottom. Those will be the erythrocytes. So whenever you see this term hematocrit, what we're talking about is erythrocytes, sometimes known as red blood cells. Then in between those layers, between the plasma and the hematocrit, it's going to be something known as the buffy coat. The buffy coat is a tiny volume of that overall aliquot of blood. The buffy coat is less than 1% of the volume. And when we look at the components of the buffy coat, we see what's in the buffy coat are cells of the immune system. It's going to be lymphocytes, B lymphocytes and T lymphocytes, macrophages, granulocytes. But there aren't very many of them. There are not all that many of them. And so they only comprise about 1% of the blood volume. The other thing we're going to find in the buffy coat are things called platelets. Platelets, we're not going to talk too much about, but the platelets are just bags. They're membrane-bound bags of vesicles, and those vesicles contain all manner of clotting factors. When platelets aren't functioning properly, we can't clot our blood properly. And if we don't have enough platelets, we're in danger of hemorrhaging. And so all of our lives, what we're doing is there will be small tears in the blood vascular system, and we plug them with these platelets. If we don't have enough platelets, we're in danger of bleeding out through those small tears. So they're really important to us, but we're going to mostly emphasize them in terms of what they're not. They're not true cells. They're what we call a formed element. They're a formed element because basically they're a membrane-bound bag that was derived from another cell. They've been derived from a cell type called a megakaryocyte. And the megakaryocytes live in the red bone marrow, and they extend processes of cytoplasm surrounded by their plasma membranes into the blood vascular system. And then those bits of processes of the megakaryocytes break off and seal up. And when they seal up, they are now the platelets. But they don't have any of the typical machinery we think of in terms of cells. They don't have a nucleus, they don't have an endoplasmic reticulum, they can't make proteins, they're just these bags of vesicles. So that's why we can't call them a proper cell, we call them a formed element. But the other major formed element we find in the blood are these erythrocytes. We'll often refer to these as red blood cells. They're red because they're chock full of hemoglobin, and that hemoglobin has a red color. But even though we call them red blood cells, they're not true cells. And I try to avoid the term red blood cell and refer to them as erythrocytes. And we'll see the reason they're not true cells is that when we produce erythrocytes, and we're doing this hundreds of millions of cells every hour of every day that we're alive, what we're doing is we're generating these erythrocytes in the red bone marrow. They start out as hematopoietic stem cells, and then they mature into something called a proerythrocyte. And then they mature into something called, I missed it. They mature into something known as a normoblast. And through this process of going from a hematopoietic stem cell, which is a kind of garden variety cell, to a proper erythrocyte, they lose all of their protein synthesis machinery. They lose their they lose their nucleus. The normoblast ejects the nucleus. They lose their mitochondria. They lose their endoplasmic reticulum. They lose their ribosomes. So all of those things we've talked about with cells so far, this ability to have an intrinsic capacity to make proteins and keep the cell alive, that's all lost. That has implications for these cells. These guys only last about 90 to 120 days. So of the hundreds and hundreds of millions of your erythrocytes, you are turning over like around 1% of them every day. And that's a big number. And we'll talk a little bit about that. But they all have to go through this process of going from a hematopoietic stem cell to a mature erythrocyte. But as we get started here, let me start with a couple of slides. This first slide is one we've seen before, but I think it's a useful slide, mostly to emphasize the concept that the cardiovascular system is a closed system. The pump, the heart, lies at the center of it, but it's a series of tubes coming out of the heart. And those tubes don't open up into the tissues. They remain closed throughout. So we start out here, we'll go out on the left side here, into the aorta. And then we'll see in a moment this aorta branches and branches. They're showing it branching into arterioles. The arterioles then further branching into capillaries. Capillaries being a single layer of endothelial cells thick. Then that blood is carried from the capillaries into the venous side, into venules, and then back to the right side of the heart. If we follow the blood coming out of the right side of the heart, remember this is the pulmonary pump, blood's going to go out through the pulmonary artery, then it's going to go through a system of capillaries. The blood never leaves the system. It stays within the capillaries, and in the lung, across the capillaries, we're going to be absorbing oxygen into the blood and getting rid of carbon dioxide. Then the oxygenated blood is then returned back to the left side of the heart. The long and the short of this figure is that the blood never leaves those tubes. It's always in the tubes. And the next figure is to show how those tubes branch. This is just well down the system. We're seeing here something known as a fourth order arteriole branching to first order arteriole, rather branching to second, third, and then we have these tiny fourth order arterioles, which then branch into the capillaries. Anytime we're talking about these arterioles, or if we're talking about their bigger cousins, the arteries, they are all surrounded by a layer of smooth muscle. And that smooth muscle now helps to control blood flow through these tubes. And I think you can imagine here, if we could imagine right here along the third order arteriole, if we surrounded this with smooth muscle, and we use the smooth muscle to reduce the diameter of this tube, that's going to increase the resistance to flow. If we increase the resistance to flow, there are paths of lesser resistance, and the blood will flow through these other paths of lesser resistance. And that's the trick that the autonomic nervous system uses to shunt blood to different places. And we know this more or less intuitively. We know that if we go out on a warm day and we do a lot of exercising, that we're going to get flushed. Basically, our skin's going to turn red. And the reason for that is we're trying to radiate more of our core temperature away. By doing all of this muscle activity, we've raised the core temperature. And now we're going to help try to get rid of that extra temperature, that extra heat, by sweating, but also by dilating the blood vessels. So we're dilating the blood vessels that go to the skin. Our skin turns red. Similarly, on a cold day, what you'll do in order to conserve heat is you'll constrict those blood vessels going out to the surface, to the skin, and then shunt that blood back into the core to retain heat. And much of that shunting is done simply by regulating the smooth muscle at different portions of this continually branching system. Then on the other side, we now gather up the blood which has come through the capillaries. And now we return it to this branching system, which goes all the way back to the vena cava, the superior and inferior vena cava, and then into the single vena cava, into the right side of the heart. But this is also a tremendously branching system. Now, as we walk through this, one of the things I want you to recognize is that with all the branching, the surface area of those tubes gets greater and greater and greater. You have a tremendous surface area of the tubes out in that branched vasculature. And that as the blood is moving through all of these branches, and there's all the surface area, that some of that blood is going to be against the walls of those tubes. And when the blood is against the walls of the tube, there's now a certain amount of friction between the blood and the wall. And that friction is going to absorb much of the energy that was imparted to the blood when the ventricular contraction took place. And that's shown in the next slide. Again, we've seen this before in a different context. But what it's showing is the division of all this branching system all the way from the single tube leaving the left side of the heart in the form of the aorta. Then a series of these large arteries, and then smaller arteries down to arterioles. Now, here in the initial stages, this pulsatile action, the systole followed by diastole, we can see that pulsatile action in these tubes. Now, it's true there's going to be a certain amount of elasticity we'll talk about with the elastin around these big tubes. So they'll expand with that activity, and they'll then impart a little bit more force back to it as during diastole. But still, we see this pulsatile arrangement of the blood going out into the tubes. During diastole, the pressure dissipates, and we start again. And that pulsatile arrangement of increasing pressure and falling pressure, increasing pressure, can be followed all the way out into the small arterioles. That's why you can feel your pulse when you take your pulse in your wrist. You're actually measuring you're feeling that pulsatile activity of the beating of the heart out in those arteries out in your wrist. But by the time we get into the arterioles, we've lost the pulsatile activity, and we get smooth flow. We now get non-pulsatile flow of blood through those tubes. And part of that is just smoothing out that pulsatile activity. But as you also notice that there's a pressure here. This is the pressure in millimeters of mercury. And so the pressure in the system falls as we get farther and farther away from the left side of the heart. And as this pressure falls, by the time we get out into the capillaries and into the venules, in the return system, this is the low pressure return. This is the 3 to 5 millimeters of mercury. It's the low pressure return. But you notice that much of that energy is dissipated through these arterioles, because this is where we get all this branching of the system into all these fine arterioles, and the energy is being dissipated by friction against the walls. There's further drop across the capillaries. So then by the time we get into the venules and coming back, we're down to that low pressure return. Now, one of the ideas that there was a question in the first lecture, and it's an important idea that's not well characterized here, because what this is showing is pressure, and it's showing the drop of the pressure through the system. But remember, it's a closed system. We have only one series of tubes. So what goes out at high pressure, that volume going out at high pressure has to come back as the same volume as low pressure. So even though the pressures are low out here on the return trip, the flow in terms of milliliters per minute is still the same. And it has to be that way because the system is closed. If you had greater flow going out through the arterial system, excuse me, than you had flow coming back through the venous system, you'd basically run out of blood. And so because it's a closed system, even though the pressures are dropping, the rates of flow remain the same. And that takes a little bit of thinking about, because you really want to have the pressure forcing faster movement. But because it's a closed system, we have to retain high rates of flow, even though we have low pressures. So any questions up to there? Well, if not, let's think a little bit about these vessels. Let's talk about artery/arterial structure. Always there'll be the internal lining of the endothelial cells. So this is the endothelial cell lining. The endothelial cells are attached to a basal lamina. In some of the bigger arteries like the aorta, there's a fairly thick layer of connective tissue in here, but we'll label this as basal lamina. And in fact, if you're looking at the major arteries, you'll see there's a connective tissue layer in here that's thick enough that it contains a serious amount of elastin. And that elastin allows, with each beat of the heart, it allows this tube to expand and then impart a little bit of energy when it contracts back again. But this is a connective tissue layer. All of this is connective tissue. And then outside of that connective tissue layer, we'll see that there are these smooth muscle cells. So the sequence is the endothelial cells to the basal lamina to a connective tissue layer, and the connective tissue layer then is attached to smooth muscle. So this is smooth muscle. Then that smooth muscle will be innervated by a postganglionic. Sympathetic neuron. Remember, postganglionic sympathetic, we should be thinking about the release of norepinephrine. And for these smooth muscle cells to respond to the norepinephrine, we need a proper receptor. Now, in this particular case, we're going to be talking about something known as an alpha-1 norepinephrine receptor. Again, in the concepts we've covered over and over again, it's the receptor that determines what's going to happen here. And you may remember we talked about the heart, we talked about beta-1 receptors. Well, we're going to talk about smooth muscle in the lung. There'll be beta-2 receptors. It's another kind of norepinephrine receptor. Actually, it's an epinephrine receptor. So, and then here's another alpha-1 norepinephrine receptor, which we often include under the heading of adrenergic receptor. Well, these are G protein coupled receptors. So these G protein coupled receptors, this is GPCR, and the binding of the norepinephrine to the alpha-1 receptor activates that cascade of events that leads to smooth muscle contraction. Now think about what's going on here. This smooth muscle is wrapping around, snaking around this tube. When we contract the smooth muscle snaking around the tube, what's that going to do to the diameter of the tube? It's going to reduce the diameter of the tube. In a way, you can think about having something wound around a garden hose and then pulling that tight to reduce the diameter of the garden hose. If we reduce the diameter of the garden hose, what are we going to do about with flow through that garden hose? Well, we're going to reduce it. So we're going to do the same thing here. By constricting the smooth muscle, by contracting the smooth muscle, leading to smooth muscle contraction, this now in turn leads to reduction in blood flow. But that reduction in blood flow is going to be local, because remember, we just said it's a closed system. So even if we shunt blood to one region, we have to shunt it to someplace else. So we're going to have a continual volume of flow through the system. But locally, what we can do is we can locally what we can do is we can decrease the diameter of the tube and reduce the flow through it. Well, we can formally describe that. We can formally describe that in something we've talked about earlier when we talked about the coronary vasculature, and that's Poiseuille's law. And Poiseuille's law is just an equation. It says the flow is equal to differential in pressure times pi times the radius of the tube raised to the fourth power divided by 8 eta L. When we think about this for our purposes, what this boils down to is the flow is proportional to the radius raised to the fourth power. The reason we can simplify that is because we're going to think about a fixed length. Put your phone away, I'd appreciate it. We're going to think about a fixed length of the tube. Eta is going to be the viscosity of the fluid in the tube. Eta is simply an integer. Delta P is the pressure differential along the length of the tube. Pi is a constant. The radius is now what we're going to vary. So this is the variable. Yes?
So when we reduce blood flow, does the pressure not change?
When we reduce blood flow, let me see, if we reduce blood flow, the pressure in the local region will increase locally. But by and large, we're going to say here that small changes in the radius, what this leads us to thinking is that small changes in the radius yield large changes in flow. We know this more or less intuitively. And we know it because we can compare a small tube with a large tube. In that tube, there's going to be a fluid. Now, if you think about it for a little bit, you'll recognize that if there's a much larger volume of fluid in this tube than there is in this tube, then the proportion of the fluid that's against the wall is going to be much less in this big tube than in this small tube. In other words, if this is a small volume inside a small tube, there's going to be a larger proportion of that fluid against the wall than there is in this larger volume in the much bigger tube. Well, the reason that's important is that when we're pumping fluid through the tube, where the energy is being dissipated is in the friction of the fluid against the wall. And if you take a course in fluid dynamics, and those of you in engineering will have to do that, one of the things you'll recognize is that if you increase the diameter of the tube, you can get way more flow through it. You all know that. And the reason for that is, if you look at the mathematics, as you get closer and closer to the wall, you end up with an infinitely narrow rim of fluid against the wall that doesn't move because of infinite friction. But as you move from the wall into the middle of the tube, because less and less of the fluid has interaction with the wall, then the fluid in the middle of the tube has almost no friction on it, and it's easy to move. So if you get a great big tube, then you have a fairly small proportion of that fluid which is interacting with the wall with high friction. But if you make a small tube, you have much greater proportion of that fluid against the wall of the tube. It's harder to get the fluid through the tube. We know this intuitively. I mean, we've all done this sort of experiment. You go to the bar and you order a drink. You order a cocktail. And the cocktail comes, and it comes with one of those silly stirring straws. But you hold it up to the light and you see, it's got an opening in it. It's a straw. I'm going to try to drink my drink through that straw. And so you put the straw into your mouth and you drop your tongue to make a vacuum. So there's a higher pressure in the outside world than in your mouth because it's the way straw works. But you can't get anything through the tube because it's a tiny tube and there's an enormous amount of friction between the drink you're trying to drink and the wall of the tube. So get a bigger straw. And that solves the problem. And it's just all that idea of the friction against the walls. But when we bring that back to the concept here, what this is telling us is that small changes in a radius of a tube have big changes in flow. And if we bring that back to this figure, then we can see how we can shunt on the arterial side, if we constrict this tube, then by constricting this tube, we increase the friction along the walls, R to the fourth. That means less blood is going to flow into this capillary bed, and you're going to now shunt blood to a path of lesser resistance, and that will be through these tubes going to different capillary beds. So that's one of the ways we regulate where blood's going to flow in this closed system, by activating smooth muscle in different regions of the arterial system to shunt blood to different places. So questions about that? So if no questions there, we'll start into blood. So let's go on to blood and talk a little bit about what are the components of blood? What's blood made of? And on a gross level, how can we sort that out? Well, we've all done this experiment. We've all gone to the doctor's office, and the phlebotomist comes out, sticks a needle in our arm, and withdraws, a few mils of blood. And then the phlebotomist disappears. Sometimes that tube is going to be sent off to a laboratory someplace. In other offices, they have the facilities to take the measurements right there. But the first thing they're going to do is they're going to separate that blood into its component parts. And that separation occurs by spinning that blood down in a centrifuge. So imagine over here that we have this tube. We imagine the tube is full of blood. Then we take that tube and we put it in a centrifuge. And we put it in a centrifuge. We're now going to spin it. And when we spin it, we're going to generate forces in this direction down on that fluid. When we generate the forces down on that fluid, we're going to separate the components of blood such that the densest try again. The densest doesn't look right. The densest components go to the bottom. Well, what's going to be the densest component of the blood is going to be the erythrocytes. Erythrocytes are denser than other cells because each erythrocyte contains about 250 million hemoglobin molecules. And these 250 million hemoglobin molecules each contain 4 iron atoms. So that means each erythrocyte contains about 10 to the 9th iron atoms. Those billion iron atoms make those cells denser than other cells. So because of that density, these erythrocytes are spun to the bottom of the tube. Immediately above the erythrocytes is going to be a layer known as the buffy coat. The buffy coat contains platelets. These are essential for blood clotting. They also contain cells of the immune system. Now, what we'll see here in a moment is these cells of the immune system are true cells. They're true / complete cell / complete cells. And then above that is this region we call the plasma. And the plasma is mostly a protein solution in water. And as such, it's about 90% water. And it's about 8% protein. And about 2% other stuff. Now, don't memorize those numbers, but recognize that basically what we have is a protein solution. And that's the least dense of all the components, so it goes to the top. Down here we have the erythrocytes that are the most dense, and they go to the bottom. But if we look at the proportion of the volume of each one of these layers, we see that the erythrocytes are about 45%. This number will vary a little bit. Maybe as low as 42, maybe as high as 46. It varies from individual to individual. Women tend to have slightly lower hematocrits than men because of the loss of blood every month with the menstrual cycle. But nonetheless, more or less, 42 to 46% of the blood is erythrocytes. Less than 1% is in the buffy coat. Which in round numbers leaves the plasma as being 55% of the volume. So those are basically the overall components of blood. We can split the blood into these three major parts. And we're going to drill down into the immune system cells a little later. And today we're going to spend a little bit of time on the platelets and the erythrocytes. One of the reasons we'll go directly to the platelets and the erythrocytes is that they constitute a part of the blood which we talk about somewhat differently. And the reason we talk about them differently is they're not true cells. They don't have all of the components of a cell that we've talked about over and over again in here. And so the euphemism we use for platelets and erythrocytes is this term formed elements. So we'll talk about the formed elements of platelets and erythrocytes. Now, the other name for erythrocytes is the red blood cells, one we use all the time. But it's not a very accurate term, because when we talk about a red blood cell, that implies that it's a proper cell, but it's not. The erythrocyte is not a true cell. It lacks a nucleus. It lacks a nucleus. It lacks mitochondria. It lacks ribosomes. It lacks a complete endoplasmic reticulum. Fundamentally, all of the parts of a cell we associate with eukaryotic cells are gone. And this is a function of the way these cells develop. They develop in the red bone marrow. So these guys all come from the red bone marrow. And they start out as something known as an hematopoietic stem cell. Now, these totipotent hematopoietic stem cells can develop, given the right growth factors, into any cells of the blood system. They can grow into erythrocytes, they can grow into all the cells of the immune system, they can grow into something called megakaryocytes, so they can go in any of those directions. But if they get the right growth factors, these hematopoietic stem cells will now mature into a cell type called a pro-erythrocyte. So the pro-erythrocyte is this hematopoietic stem cell that's on its way to being developed into a proper erythrocyte. The pro-erythrocyte then gives rise to something called a normoblast. Now, if you look up on Wikipedia or someplace and you look up the development of erythrocytes, you're going to see many, many more steps in here. There are lots and lots of steps which have been, over the years, categorized by the change in different staining techniques. But what I want you to hold on to is we have a pro-erythrocyte which can receive growth factors. The binding of growth factors to pro-erythrocytes then is going to tell these cells to divide and further differentiate into a normoblast. The normoblast ejects its nucleus. That's a pretty serious thing to do. Remember, all of the genes that are going to instruct the cell how to make proteins, those are all in the nucleus, so that's gone. So this cell no longer can no longer make types of RNA to make protein. Someplace in this process, we're also going to eject ribosomes. And we're also going to eject mitochondria. In this process of development, we are basically biasing the cell to produce hemoglobin, such that by the time we get done with this thing, we're going to find that an erythrocyte is hardly anything more than it's a bag of hemoglobin. Effectively, we have biologically, evolutionarily, made the decision to make these highly specialized cells whose almost sole function is to carry oxygen on hemoglobin, and to get the maximum number of these hemoglobin molecules into this tiny cell, which we're going to see is no more than about 3 micrometers in diameter, about 7 to 8 micrometers wide. We've gotten rid of everything else. We've just sacrificed all those organelles. There's an upshot of this, which is that erythrocytes have a limited lifespan. Typically, an erythrocyte that's born today in your red bone marrow, this lifespan is going to be something between 90 and 120 days. Effectively, they wear out, and those proteins that are worn out can't be replaced. These erythrocytes have gotten rid of their protein synthesis machinery, so they only last about 90 to 120 days. Telling you that every minute of every day, or basically every day, you're turning over about 1% of all your erythrocytes. It's an enormous number. Tens and tens of millions of these cells are being produced every minute of every day. But that's simply because they don't live very long. The other formed element I'll tell you about, let me go back. Let me go to the backboard. The other formed element is a platelet. And the platelet is derived from a cell type called a megakaryocyte. But these are formed elements because in the end, there are these tiny bags of vesicles with clotting factors. They're essential for blood clotting. And where they come from in the last couple minutes is that there are going to be capillaries. This is meant to be a capillary in the red marrow. And then there will be erythrocytes flowing through the capillaries. But up here in blue is going to be a megakaryocyte. And what it does is it sends a process of its cell down here between the endothelial cells. And then this bit of the process of the megakaryocyte will break off. And when it breaks off, this is now a platelet. If you look at the cytoplasm of these megakaryocytes, they have all these vesicles in them that contain the clotting factors. But down here, they're living right beside the blood vessels, the red bone marrow, extend a process in, and that breaks off as a platelet. These are considered to be formed elements. They're not true cells. They're just bags of cytoplasm of the megakaryocyte surrounded by a bit of its plasma membrane that contain the clotting factors, which are essential to keep us alive, but we'll leave the platelets there. Hi.
A quick question.
Sure.
So, if normoblasts eject their nucleus, they don't have any DNA.
They're done with their DNA.
So, how do you identify, like, at a crime scene, like at blood? How do you identify people?
There'll be other cells around. You can't, if you simply have, well, even if you have blood, you'll have the Buffy coat.
Okay.
And so there you'll have proper cells. So all the cells of the immune system then can be isolated, and those then will show you the DNA of that individual. So, but the erythrocytes, you're right, they're useless for DNA analysis. But they're 1% of that blood will be cells of the immune system, and you can use those.
So there's a chance that a blood sample won't be viable.
I suppose there's a small chance that if you don't have enough blood, that you don't get enough true cells that you could do a proper genetic analysis. I think that's true. Yeah.
Thank you.
Sure.
I have a two-part question. After the normoblast, is it then an erythrocyte?
No, after normoblast, it becomes something called a reticulocyte.
Reticulocyte.
I'll do that tomorrow, or Wednesday. The reticulocyte gets its name because it has remnants of the endoplasmic reticulum. And when you look at it and you stain it, you see that you have these little bits of remnants of endoplasmic reticulum, and then that gives the cell a name called the reticulocyte. It's actually important clinically because the reticulocyte is a kind of proxy measure for how fast you're turning over erythrocytes. And if you have high reticulocyte count, it indicates you probably have a bleed someplace and you're turning over blood faster. And so they're going to start looking for where that bleed is.
Cool. And my second question is,